Hi, welcome to Alab Science's flow cytometry classroom. This session aims to introduce the fundamentals of flow cytometry panel design. You will gain a basic understanding of why panel design is crucial in flow cytometry, what to consider when performing panel design, and how to execute it effectively. When we talk about panel design, it is because our flow cytometry antibodies are conjugated with fluorophores. This video primarily guides you through the concepts of light, fluorescence, and fluorescence spectra. We all know that light exhibits wave-particle duality. The wave-particle duality of light means that light possesses both wave-like and particle-like properties. Light has wave properties, characterized by peaks and troughs. Displayed here is a spectrum of waves in nature. We can see that the 7 colors, red, orange, yellow, green, cyan, blue, and violet, are the visible light that the human eye can see, occupying a very small range in the spectrum. The wavelengths of visible light decrease progressively from red to violet, meaning violet light has the shortest wavelength, while red light has the longest. Fluorescence refers to a type of photoluminescence where a substance, when exposed to excitation light of a certain wavelength, absorbs the light energy, enters an excited state, and then quickly returns to the ground state by emitting light of a longer wavelength than the excitation light. Many fluorescent substances cease to emit light immediately once the excitation light stops, a property known as fluorescence. Fluorescence involves 2 key elements, excitation light and emission light. Based on the excitation and emission light, we can plot fluorescence spectra, where dashed lines represent excitation light and solid lines represent emission light. In the spectrum, the peak of the excitation light corresponds to the excitation max, which in turn correlates with the emission max on the emission spectrum. The emission light covers a relatively broad range in the spectrum. As can be seen from the spectrum, each of fluorescence emission spectrum is a larger scope. Consequently, we can see that the emission spectra of these fluorophores overlap. Due to this overlap, when a flow cytometer detects fluorescence emitted by antibody-conjugated fluorophores, it may detect adjacent fluorophores. Given that the available fluorophores for flow cytometry are limited, multicolor experiments often encounter fluorophore overlap. Therefore, it is necessary to use appropriate panel design to minimize cross-color interference. That's all for today's session. In our next session, we will delve deeper into fluorescence spectra and the principles of flow cytometry detection, helping you understand the characteristics and principles of fluorescence compensation. Please scan the QR code to follow Alab Science on YouTube or search for Alab Science on TikTok for more flow cytometry knowledge. Let's unlock more together.